Welcome back to the deep dive. Today, we are getting into something really fundamental, adaptive immunity.
Right.
And this isn't just, your basic defense mechanism. We're talking about a system so precise, it's actually revolutionizing how we treat things like cancer.
That's absolutely the key point. It's a game changer. Think about diseases like malignant melanoma. Historically, outcomes were, well, pretty grim.
Right.
And for a long time, the immune connection was sort of seen indirectly. Chronic inflammation may be driving liver cancer from Hep B or C.
Or those older sort of cruder methods.
Exactly, like using the TB vaccine strain, BCG, for bladder cancer.
hmm
Helpful, but not exactly precision targeting.
But now things are shifting dramatically. Our sources really highlight two major modern approaches. First, immunomodulation,
Chemical signals, basically.
using these monoclonal antibodies, MABs, to essentially release the brakes on the immune system's T cells.
Yeah, either stopping signals that inhibit them or providing signals that stimulate them, like taking the foot off the brake or pushing the accelerator.
And these combinations are actually working.
They really are. We're seeing melanoma patients live much longer, some even achieving cures just by letting their own T cells get back into the fight.
Okay, that's impressive. But the second one, adoptive cellular therapy, ACT, that sounds almost like science fiction.
It does, doesn't it? But it's real. This is where you take a patient's own T cells out of their body,
Uh-huh.
genetically engineer them in the lab to express these things called chimeric antibody receptors, CRs.
Creating superkiller cells.
That's exactly what they are. You're essentially giving the T cell a new targeting system, an antibody fragment, that binds super tightly to a specific marker, often on a cancer cell.
And the results.
They can be stunning, especially in things like B-cell acute lymphocytic leukemia. We're talking complete remission rates in the 70 to 95% range.
Wow.
In patients who often had run out of other options, it's truly remarkable.
That success really underscores that adaptive immunity isn't just a general defense. It's specific, it remembers, it's deeply integrated, and crucially, it seems like we can even program it.
Precisely. And to understand how we can engineer things like CRT cells, we need to understand the natural system first.
Okay, so what defines this adaptive system? How is it different from, say, our innate immunity?
Well, there are four key characteristics. First, discrimination. It's incredibly good at telling your own body cells from non-self invaders or altered cells.
Highly selective.
Extremely. Second, specificity. An adaptive response is usually directed against one particular target, one specific antigen.
Okay.
Third, diversity. The system has this almost unbelievable capacity to recognize trillions, literally trillions, of unique molecules.
Trillions. How? We'll have to get into that.
We will. And finally, memory. This is huge. Once you've encountered a specific threat, the adaptive system remembers it, allowing for a much faster, stronger response the next time.
That's why vaccines work, and why you usually only get chickenpox once.
Exactly. And this whole system operates through two main branches, functionally speaking.
Right, the two arms.
You have humoral immunity. This is mediated mostly by B cells, which produce antibodies that circulate in your blood and fluids.
The antibody factory branch.
Sort of, yes. And then you have cellular immunity, or cell-mediated immunity. This is the domain of the T cells.
Including those killer cells we mentioned.
Yes, the cytotoxic T lymphocytes, or CTLs, that do the direct killing, but also the T helper cells that, well, orchestrate the whole response.
Okay, let's start with the trigger. The thing the immune system actually recognizes. We call it an antigen. What exactly is an antigen?
So an antigen, sometimes called an immunogen, is really any substance, usually a protein or a polysaccharide, that can provoke an immune response.
But the immune system doesn't grab the whole molecule, does it?
No, that's a key point. It recognizes specific small regions on that antigen. These regions are called epitopes, or antigenic determinant sites.
Ah, so the epitope is the actual binding site?
Precisely. Think of the antigen as the whole enemy ship, and the epitope is the specific target site, like an exhaust port that your weapon locks onto.
Got it. And the number of these epitopes on an antigen matters too, right? Valence?
Yes, valence. Antigens with multiple epitopes, multivalent antigens, tend to elicit a much stronger immune response because they can cross-link more receptors.
That makes sense. It also helps explain things like haptens, these tiny molecules like penicillin. They aren't big enough on their own.
Exactly. Haptens are too small to be immunogenic by themselves. They only trigger a response when they attach to a larger carrier molecule, often one of your own proteins.
And that's why some people develop allergies to drugs. Their immune system sees the drug-protein complex as foreign.
That's often the mechanism, yes. The hapten makes the self protein look non-self.
Okay, so we know what triggers the response. How do we actually get this immunity? Our sources lay out four main ways.
Right, and we can categorize them based on two factors. Is it natural or artificial? And is your body actively making the response or just passively receiving protection?
Let's start with natural. What's naturally acquired active immunity?
That's the most common way. You get infected with something, measles, the flu, whatever. Your body actively fights it off and, crucially, builds memory T cells and B cells. This immunity is usually long-lasting.
Okay, so infection leads to active, long-term protection. What about the passive natural route?
That's the transfer of antibodies from mother to baby. It happens across the placenta during pregnancy and through breast milk after birth.
Ah, so the baby gets pre-made antibodies.
Exactly. It provides vital protection while the baby's own immune system is still developing. But it's passive. The baby didn't make those antibodies, so it's temporary, usually fading after about 6 months.
A temporary shield. Makes sense. Now, the artificial ways.
They mirror the natural ones. Artificially acquired active immunity is basically vaccination.
Right. We introduce a harmless version or piece of the pathogen.
Yes, like a killed virus, an attenuated, weakened one, or just specific antigens. The point is to intentionally expose your body to the foreign material so it actively builds its own memory response without causing disease.
So vaccination is active, artificial immunity. And the last one, artificial passive.
This is when you receive preformed antibodies from another source. For instance, getting an injection of antitoxin if you've been exposed to botulism. The antitoxin antibodies come from, say, an immune horse or human donor.
So you get immediate protection,
But your body didn't make them, so it doesn't build memory. The protection is temporary. The antibodies eventually get cleared out.
Okay, that framework makes sense. Now let's dive into the core mechanism, especially for T cells. How do they tell self from non-self? You mentioned MHC molecules earlier.
Yes, the Major Histocompatibility Complex, or MHC. In humans, we often call it the HLA system, human leukocyte antigen. These molecules are absolutely critical for T cell recognition. They're like the display platforms for antigens.
And there are two main types, Class I and Class II. And they present different kinds of information, right?
Exactly. They direct the whole cellular response based on where the antigen came from. Let's start with MHC Class I.
Okay, MHCI. Where do we find these?
You find MHC Class I molecules on the surface of almost every single nucleated cell in your body. Pretty much everything except red blood cells.
So they're everywhere. What do they display?
They're involved in what's called endogenous antigen processing. They bind to peptides, small protein fragments, that originate from inside the cell's cytoplasm.
Inside. Like what?
Well, in a healthy cell, it's just displaying fragments of normal self-proteins. It's like saying, everything's fine in here. But if the cell gets infected with a virus, or if it becomes cancerous and starts making abnormal proteins.
That it displays those viral or cancer peptides in MHCI.
Precisely. It's basically sending up a red flag. I'm compromised internally.
And who sees that flag?
That flag is recognized by CD8 plus T cells, the cytotoxic T lymphocytes, CTLs. Presentation in MHC essentially marks that cell for destruction by CTLs.
The internal distress signal leads to elimination. Got it. What about MHC Class II?
MHC Class II is different. It's much more restricted. You only find it on specialized antigen-presenting cells, or APCs.
Which are?
Primarily macrophages, dendritic cells, and B cells. These are the cells whose job it is to patrol tissues and engulf foreign material.
So MHCT is involved in processing stuff from outside the cell.
Exactly. It's called exogenous antigen processing. The APC engulfs something, like a bacterium or some foreign debris, breaks it down internally, and then displays fragments of that external invader on its MHC Class II molecules.
Okay, so Class I is what's wrong inside me. Class II is what I found out there. Who recognizes Class II?
MHC Class II presentation is recognized by CD4 plus T cells, the T helper cells.
The orchestrator.
Right, so MHCI presenting an internal threat leads to killing by CD8 cells. MHC2-XE presenting an external threat leads to activation of CD4 helper cells, which then coordinate the broader immune response, like telling B cells to make antibodies.
That distinction is crucial. So these T cells, CD4 and CD8, they don't just appear ready to go, right?
Yeah.
There's a development
Oh,
progress.
absolutely. T cells originate in the bone marrow, but then they mature in the thymus. This is a critical stage called thymic selection.
Selection. What gets selected?
Well, it's more about what doesn't get selected. About 98% of the T cells that enter the thymus actually die there through apoptosis.
And 98% why?
Mostly because they fail positive selection, can't recognize MHC properly, Or, crucially, they fail negative selection, meaning they react too strongly to self-antigens presented in the thymus. The body eliminates these potentially self-reactive cells. It's a key part of central tolerance.
So only the T cells that can recognize MHC but don't strongly attack self are allowed to mature and leave the thymus.
Exactly. These mature cells are then considered naive. They're ready, but they haven't encountered their specific antigen yet.
Okay, so now we have these naive T cells circulating. How do they get activated when they finally meet their antigen on an APC? It's not just simple binding, is it?
No, definitely not. Full T-cell activation requires 3 distinct signals. It's a critical safety system to prevent accidental activation.
3 signals. Okay, what's signal 1?
Signal 1 is recognition. This is when the T-cell receptor, the TCR, on the T-cell's surface physically binds to the specific antigen peptide being presented by the MHC molecule on the APC. CD4 binds MHCDI, CD8 binds MHCI. This forms what's called an immune synapse.
Okay, the specific handshake, what's signal 2?
Signal 2 is co-stimulation. This is like a safety check. It involves other matching pairs of molecules on the APC and T-cell surfaces. The most well-known pair is the B7 protein, also called CD8 on the APC binding to the CD28 receptor on the T-cell.
And why is this co-stimulation so important?
Because if a T-cell receives signal 1, recognition, without signal 2, co-stimulation, it doesn't get activated. Instead, it becomes anergic.
Anergic, meaning?
Basically, turned off, unresponsive. It recognizes its target, but it can't act. It's a crucial peripheral tolerance mechanism to shut down T cells that might react to self-antigens presented by cells that aren't activated APCs, providing co-stimulation.
Wow. So recognize without confirmation equals shut down. That's a powerful safety feature. What's signal 3 then?
Signal 3 comes from cytokines. These are signaling proteins released by the APC and sometimes the T cell itself once signals 1 and 2 are go. These cytokines guide the T cell's fate, telling it to proliferate, multiply, and differentiate into the right type of effector cell or a memory cell.
And this differentiation is where we get those different types of T helper cells, the CD4 ones.
Exactly. Depending on the cytokine environment, signal 3, a CD4 T cell can become different subtypes, each with a specialized role.
Like what?
Well, you have Th1 cells. They produce cytokines like interferon gamma, which are great for activating macrophages and boosting the activity of CTLs, really important for dealing with intracellular microbes like viruses and certain bacteria.
Okay, Th1 for intracellular fights.
Then there are Th2 cells. They produce different cytokines like IL-4 and IL-5, which are crucial for stimulating B cells to make antibodies, especially IgE, and for fighting off parasitic worms, helminths.
Th2 for antibodies and parasites. Any others?
Yes. Th17 cells are important too. They respond to certain bacterial and fungal infections, mainly by producing IL-17, which recruits neutrophils, those rapid response inflammatory cells, to the site of infection. They also boost production of antimicrobial peptides like defensins.
Th17 for recruiting inflammation fighters. That covers a lot of ground.
It does, but there's one more crucial CD4 subset. Regulatory T cells, or TREGs.
Ah, you mentioned these. They calm things down.
Yeah, precisely. Their job is immune homeostasis. They produce anti-inflammatory cytokines like IL-10 and TGF-beta. They actively suppress the activation and function of other effector T cells. They're essential for preventing autoimmune reactions and dialing down the immune response once an infection is cleared. The peacekeepers.
Absolutely critical. So while the helper cells, CD4, are directing traffic and specializing, the activated CD8 cells become the actual killers, the CTLs. How do they kill?
They have a couple of main methods. Once a CTL recognizes its target, a host cell displaying a foreign peptide in MHC Class I or alive, it binds tightly. Then it can use the perforin pathway. It releases proteins called perforins that punch holes in the target cell membrane.
Like poking holes in it.
Exactly. And through those holes, it injects enzymes called granzymes, which trigger apoptosis-programmed cell death inside the target cell. It's a controlled demolition.
Okay, performed in granzymes. What's the other way?
The other main route is the Fas-Fasi pathway. The CTL expresses a protein called Fas ligand, Fas-LL, on its surface. If the target cell has the matching Fas receptor, which many cells do, this binding directly triggers the apoptotic pathway within the target cell, another way to induce clean self-destruction.
Efficient and relatively clean ways to eliminate compromised cells.
Okay.
That gives us a good picture of the cell-mediated side. What about the other arm humoral immunity, B cells and antibodies.
Right. So B cells also have specific receptors, the B cell receptor or BCR. It's essentially an antibody molecule, usually IgM or IgD, anchored in the B cell's membrane, along with some signaling accessory proteins.
So the BCR is the B cell's antigen detector. How do they get activated?
There are two main ways, similar in concept to T cell activation needing help. The most robust way is T cell dependent activation.
Needs help from T cells. How does that work?
The B cell's BCR binds to its specific antigen. The B cell then internalizes this antigen, processes it, and presents fragments on its own MHC Class II molecules.
Wait, so the B cell acts as an APC too? Yes, for T helper cells. An activated TH cell that recognizes the same peptide presented by the B cell can then provide the necessary help. This involves direct contact, like CD40L on the T cell binding CD40 on the B cell, and the T cell releasing specific cytokines.
So it's like a two-factor authentication for making antibodies. The B cell finds the antigen, and the T cell confirms it's a real threat.
That's a great analogy. This T cell help, signal 2 for the B cell effectively, is crucial. It drives the B cell to proliferate massively and, importantly, to differentiate.
Into what?
Into plasma cells,
Yeah.
which are absolute antibody-secreting factories, churning out huge amounts of antibody, and also into memory B cells, which provide that long-term humoral memory. This process also involves things like class switching, changing the antibody type from IgM to IgG, IgA, or IgE, and affinity maturation, making the antibodies even better binders.
So T-dependent activation gives you high-quality antibodies, and memory was the other way. That's T-cell independent activation. This can happen with certain types of antigens, usually large polymers with repeating subunits, like the lipopolysaccharide, LPS, from bacteria.
How does that work without T cells?
These antigens can directly cross-link many BCRs on the B cell surface simultaneously, providing a strong enough signal to activate the B cell directly without T cell help.
Sounds faster. Is it as good?
It is faster, which can be useful for quick responses to common bacterial components. But the downside is that it primarily generates lower affinity IgM antibodies. You don't typically get class switching to IgG or IgA, and critically, you form very few memory B cells, so it's a quicker but less sophisticated and less durable response.
Okay, T-dependent is the gold standard for strong, lasting antibody immunity. Let's look at the antibodies themselves, immunoglobulins or Ig. What's their basic structure?
The classic antibody structure is a Y-shaped molecule. It's made of 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains held together by disulfide bonds.
And the different parts of the Y do different things.
Absolutely. The top parts of the Y, the 2 arms, contain the variable V regions of both the heavy and light chains. This is where the variation is, and these regions form the antigen-binding sites, called the Fab fragments. The sequence here determines the antibody's specificity, what epitope it binds to.
So the tips of the Y grab the antigen. What about the stem?
The stem of the Y is formed by the constant C regions of the heavy chains. This part is much less variable within a given class of antibody. The constant region, particularly the very bottom part called the FC fragment crystallizable, determines the antibody's class or isotype, IgG, IgM, IgA, IgE, IgD, and dictates its function.
How does the FC region determine function?
Because different immune cells have receptors, called FC receptors, that bind to the FC regions of specific antibody classes. So, an IgG antibody coating a bacterium can then be recognized by an FC receptor on a phagocyte, triggering engulfment. The FC region also determines if the antibody can activate complement or cross the placenta.
Okay, structure dictates function. Let's run through the main classes and their key jobs. What about IgG?
IgG is the workhorse in your blood serum. It makes up about 80% of the immunoglobulin there. It's a monomer, a single Y shape. Crucially, it's small enough to cross the placenta, providing that natural passive immunity to the fetus. It's also great at opsonization, flagging for phagocytosis, and neutralizing toxins and viruses.
The main player in the blood, and crosses the placenta. What about IgM?
IgM is usually the first antibody class produced in a primary immune response. In serum, it exists as a massive pentamer, 5 Y-shaped units joined together by a J chain.
5 Ys together. That sounds huge.
It is. This gives it 10 antigen-binding sites, making it incredibly efficient at agglutinating clumping pathogens, and importantly, it's the most effective antibody class at activating the classical complement pathway, that cascade of proteins that helps destroy bacteria.
First responder, big clumper, complement activator. Got it. Then there's IgA.
IgA is interesting because while there's some in the serum, its main claim to fame is being the major secretory immunoglobulin. It's actively transported across mucosal surfaces lining your gut, your respiratory tract, in saliva, tears, and breast milk.
Secreted where the action often starts.
Exactly. Out there, it usually exists as a dimer, two Ys joined by a J chain and a secretory component. Its primary role in secretions is immune exclusion. It binds to pathogens and toxins right there on the mucosal surface, preventing them from even attaching to or entering your host cells. It blocks them at the gate.
Super important for barrier defense. What about the other two, IgD and IgE?
IgD is found in very small amounts in serum. Its main role is actually on the surface of naive B cells, where it acts as part of the B cell receptor along with IgM.
So mostly a receptor. And IgE?
IgE is present in the tiniest amounts in serum normally. It's mostly known for its role in allergic reactions. It binds very strongly to Fc's receptors on mast cells and basophils. When antigen then cross-links this bound IgE, it triggers these cells to degranulate, releasing histamine and other inflammatory mediators.
The allergy antibody.
Yes, but its likely intended role is probably in defense against parasitic worms, as it can also bind to eosinophils, which are involved in fighting parasites.
Okay, 5 classes, distinct structures, and functions. Now, the kinetics, how antibody levels change over time. The difference between the first time you meet an antigen and the second time is stark, right?
Absolutely. The primary response happens after first exposure. There's a lag phase, maybe a week or two, while B cells are getting activated. Then you start seeing antibody levels rise, primarily IgM first, followed by a switch to IgG, but the overall levels aren't huge, and the affinity might not be optimal initially.
Slow start, IgM first, modest peak. What happens on re-exposure?
The secondary response, or anamnestic response, is completely different thanks to those memory B and T cells generated during the primary response. The lag phase is much shorter, maybe just a few days,
Much faster.
and the antibody production is much more rapid, reaches a significantly higher peak level, and consists mainly of high-affinity IgG right from the start, though some IgM is still made. It's faster, stronger, and better quality.
That's the immunological memory in action. Phenomenal. How does the body generate the sheer diversity needed? You said trillions of specificities. How from a limited number of genes?
It's truly ingenious. There are several mechanisms, but two key ones our sources mention for antibodies are combinatorial joining and somatic hypermutation.
Combinatorial joining.
In the developing B cell, and T cell for its receptor, the genes coding for the variable regions aren't intact. They exist as segments, V, variable, D, diversity only in heavy chains, and J, joining segments. During development, DNA is literally cut and pasted randomly, joining one V, one D is present, and one J segment together.
Like shuffling and dealing gene segments.
Exactly. The sheer number of possible combinations of these segments generates enormous initial diversity even before the B cell sees antigen.
Okay, that creates a huge starting pool. What's somatic hypermutation?
This happens after a B cell has been activated by antigen and is receiving T cell help, usually in specialized areas of lymph nodes called germinal centers. The genes encoding the variable regions undergo an incredibly high rate of point mutations, tiny changes in the DNA sequence.
Mutations, isn't that usually bad?
Normally, yes, but here it's targeted specifically to the antibody V region genes. Some mutations will make the antibody bind worse to the antigen, and those B cells will likely die off. But some mutations, purely by chance, will make the antibody bind better, higher affinity or avidity.
Ah, so it's like accelerated evolution in the B cell population.
Precisely. B cells with improved receptors get selected because they compete better for antigen and T cell help, so they proliferate more. This process fine-tunes the antibody response, leading to the production of very high-affinity antibodies as the immune response progresses.
Incredible. Okay, so we have these specific high-affinity antibodies. How do they actually stop the pathogen? What are their main actions?
We touched on some, but let's consolidate. A major one is neutralization. The antibody simply binds to the pathogen or toxin in a way that blocks its harmful activity.
Like how?
Like secretory IgA binding to bacteria on your gut lining, preventing them from adhering. Or IgG binding to a virus particle, preventing it from docking with and entering your host cell. Or antibodies binding to a bacterial toxin, preventing it from reaching its cellular target. Simple blockade.
Physical interference? What else?
Opsonization. This means to make tasty. Antibodies, particularly IgG, coat the surface of a pathogen. Phagocytic cells, like macrophages and neutrophils, have FEC receptors that bind the stem, FEC region of these bound antibodies. This provides a much stronger eat-me signal, enhancing phagocytosis.
The antibody acts like a handle for the phagocyte to grab onto.
Exactly. And another mechanism is immune complex formation. Because antibodies have at least 2 binding sites, and IgM has 10, they can cross-link multiple antigen molecules or even whole cells together.
Clumping things up.
Yes. If the antigens are on cells, like bacteria, it's called agglutination. If the antigens are soluble molecules, like toxins, it's called precipitation. These large complexes are generally easier for the immune system to clear, often via phagocytosis or the complement system.
Makes sense. But can that cause problems?
It can. If too many immune complexes form or they aren't cleared efficiently, they can get deposited in tissues like the kidneys or blood vessel walls and trigger inflammation. That's the basis of type 3 hypersensitivity reactions.
Okay. So this incredibly powerful, specific, adaptable system, it sounds like it needs incredibly tight control.
Yeah.
Because if it makes a mistake.
Exactly. The flip side of this power is the potential for disaster if it attacks the wrong target, namely self. This brings us to immune tolerance.
Tolerance meaning not attacking yourself.
Precisely. Acquired immune tolerance is the state of unresponsiveness to a specific antigen, particularly self-antigens. It has to be learned and maintained.
How does the body learn tolerance?
There are 2 main levels. Central tolerance happens during lymphocyte development. We already mentioned negative selection in the thymus, where T cells that strongly recognize self-antigens are killed off. Apoptosis. A similar process happens for B cells in the bone marrow.
Removing the threat before it even gets out.
That's the goal. But it's not perfect. Some self-reactive lymphocytes inevitably escape into the periphery.
So there must be backup mechanisms.
Yes, that's peripheral tolerance. This involves mechanisms operating outside the primary lymphoid organs. We talked about energy-inducing unresponsiveness in T cells that recognize antigen without co-stimulation. Another key player here are those regulatory T cells, TREGs we discussed, which actively suppress self-reactive lymphocytes they encounter in the tissues.
Multiple layers of safety checks. But sometimes they fail.
They can. And when tolerance fails, or when the immune system mounts an inappropriate or exaggerated response to a harmless foreign antigen, we get immune disorders.
Like hypersensitivities.
Right. Hypersensitivities are basically immune responses that cause tissue damage. The Gelkum's classification divides them into 4 types based on the mechanism.
Okay, type I.
Type I is immediate hypersensitivity. This is your classic allergy, hay fever, food allergies, bee sting anaphylaxis. It's mediated by IgE antibodies.
How does IgE cause that?
On first exposure, sensitization, you make IgE against the allergen, and this IgE binds to mast cells and basophils. On subsequent exposure, the allergen cross-links the IgE on these cells, causing them to rapidly release histamine and other inflammatory mediators, leading to the allergy symptoms.
Instant reaction triggered by IgE. What's type 2?
Type 2 is cytotoxic hypersensitivity. Here, IgG or IgM antibodies are mistakenly directed against antigens on the surface of your own cells or tissues.
Attacking your own cells.
Yes. Binding of these antibodies can lead to cell destruction via complement activation or antibody-dependent cell-mediated cytotoxicity, ADCC. Classic examples are mismatched blood transfusions where antibodies attack the foreign blood cells or hemolytic disease of the newborn due to Rh factor incompatibility.
Okay. Antibodies attacking cell surfaces. Type 3.
Type 3 is immune complex hypersensitivity. We mentioned this possibility earlier. It happens when there's an excess of antigen and IgG, or IgM antibodies, leading to the formation of large quantities of immune complexes that aren't cleared properly.
The complexes build up.
Yes, they deposit in places like small blood vessels, kidney glomeruli, and joints. This deposition triggers complement activation and inflammation, leading to tissue damage. Diseases like serum sickness or certain aspects of lupus involve type 3 mechanisms.
Okay, antigen clumps causing trouble. And type Thoi.
Type Phi is different because it's not mediated by antibodies. It's cell-mediated hypersensitivity, driven primarily by T cells, both helper T cells and CTLs. It's also called delayed Tuck hypersensitivity, DTH, because the reaction typically takes 24 to 72 hours to develop.
Why the delay?
Because it takes time for the relevant T cells to migrate to the site, recognize the antigen presented by local APCs, become activated, and recruit other inflammatory cells like macrophages. The classic example is the reaction to a tuberculin skin test. TB test. Also, allergic contact dermatitis, like the rash you get from poison ivy, is a type 5e reaction mediated by T cells reacting to chemicals from the plant bound to skin proteins.
Okay, 4 types, different mechanisms. And when tolerance completely breaks down against self-antigens, that leads to
Autoimmune diseases. This is where the immune system loses self-tolerance and mounts a sustained attack against the body's own components. Things like rheumatoid arthritis, attacking joints, systemic lupus erythematosus, attacking DNA, nuclear proteins, etc. Type 1 diabetes, attacking insulin-producing cells.
what causes this loss of tolerance?
It's usually complex and multifactorial. Genetics play a role. Certain HLA types predispose individuals. Environmental triggers are often involved, potentially infections, molecular mimicry, where a pathogen antigen resembles a self-antigen, or tissue damage releasing hidden self-antigens. And often there seems to be a failure in regulatory mechanisms, like insufficient TREG function.
A complex failure leading to self-attack. The MHC system is also central to another immune problem, right? Transplantation.
Absolutely. MHC molecules are the primary antigens recognized as foreign during transplantation rejection. Because MHC genes are so polymorphic, variable between individuals, the recipient's immune system almost always sees the donor organ's MHC molecules as non-self.
So the recipient attacks the graft.
Yes, that's host-versus-graft disease. The recipient's T cells, particularly CTLs, recognize the foreign MHC on the donor cells and attack the transplanted organ. This is why immunosuppressive drugs are essential after most organ transplants.
Is the reverse possible?
Yes, particularly in bone marrow or hematopoietic stem cell transplants. Here, you're transplanting immune cells themselves. If these transplanted immune cells are immunocompetent, they can recognize the recipient's entire body, all their MHC molecules, as foreign.
So the graft attacks the host.
Exactly. That's graft versus host disease, GVHD. And it can be very severe, affecting skin, liver, intestines, and other tissues. Careful matching of HLA types between donor and recipient is critical to minimize both types of rejection.
It really highlights how central MHC is to distinguishing self from non-self. So bringing it all together, thinking back to those HER-T cells curing leukemia, it's really about harnessing the specificity and power we've just discussed.
It absolutely is. Adaptive immunity hinges on that incredibly specific recognition by T cells and B cells, guided by MHC presentation, and executed through diverse mechanisms like antibody production with class switching, affinity maturation, and targeted cell killing by CTLs.
The detail is incredible, from combinatorial joining creating diversity to TREGs maintaining peace.
And the key takeaway, I think, is the sheer power balanced on a knife edge. This system can eliminate deadly cancers when properly directed. It can give you lifelong protection from pathogens after a single encounter or vaccination. But that same power, if misdirected due to a failure in tolerance or regulation, leads to devastating chronic autoimmune diseases or potentially fatal hypersensitivity reactions.
It seems like the difference between a cure and a catastrophe can sometimes boil down to whether a single receptor binds or fails to bind correctly, or whether a single co-stimulatory signal is delivered or not.
That's the exquisite precision and the inherent risk of adaptive immunity. It's a system of phenomenal power, operating with remarkable but not infallible control. It's the constant tightrope walk happening inside us all the time.
A fascinating and vital system. Thank you so much for walking us through it, and thank you for joining us on the Deep Dive.